My name is Bob Bazell, and it's been my great pleasure to have been paying attention to the research into Huntington's disease since the early 1980s. And it's a great honor to be able to come back and talk to you about astounding progress that's been made and the prospect of even more exciting things very soon to come. This is a very, very exciting time. I was a science and medicine reporter for NBC for many years, and I remember just the excitement that by studying the Venezuelan group that the gene would be discovered, and that was an enormous achievement. It leads to everything that you're about to hear follows from that. But I can't emphasize enough how much, after many, many years of just talking about potential things, that we are actually going to start with a physician who is giving a drug to human beings with Huntington's disease. And that is astounding. And it's just the first step. So that physician is Dr. Edward Wild. I pronounced that correctly. Yes, good. He's the principal researcher at University College London Huntington's Disease Center and a consultant neurologist at the National Hospital for Neurology and Neurosurgeon, Queen Square in London. He has a lot more credits. You can read about them in this program. I'm not going to say anything else except to say that he is putting drugs into human beings, not mice, not worms, but human beings with Huntington's disease. Doctor.
You do the thing with the microphone. Can you hear me? Can you hear me if I sniff? Is that okay? Okay, good. So, hi everyone. It's a surpassing honor to be here. I feel too young and too inexperienced to be here, having only been in the field for 12 years. I'm going to be presenting a lot of excitement that exists because of a huge amount of collaborative effort over many decades by many hundreds of people in which the HDF has been instrumental from the outset. The title of my talk is A Little Gale, and if you know all of Shakespeare off by heart, you'll know why, but don't worry, I'll come back to it at the end. So I'm going to start with the basic dogma of biology, because that's always a good place to begin. And everything you need to know about biology is that our genes contain recipes for proteins, and proteins are the chemical entities that do stuff in our cells. All of the important things in our cells is done by proteins. Every gene is, broadly speaking, a recipe for one protein, but the genes are very precious and very well protected. So like a precious recipe book that's been handed down through the generations, we don't take that book into the kitchen, we take copies of it whenever we want to use one of the recipes. So we make a copy, and that copy is the message molecule. It's made of something called RNA, which is a bit like DNA, but it only has one strand. And that RNA is what is then used to make many copies of the protein. This protein, this pink potato blob, is an electron microscope rendition of the mutant Huntington protein. And it's only in the past 12 months that we've been able to actually look at a molecule of mutant Huntington and say, this is what it looks like. It looks like a moldy potato. So this is public enemy number one in Huntington's disease. It is the cause of all damage in the Huntington's disease brain and body. If we could stop that protein from forming, Huntington's disease would not happen. So, once it's there, a ton of other stuff goes wrong. Every one of these red blobs, red boxes, is something that researchers are working on somewhere as a means of treating one of the consequences of the presence of this protein. However, and there are lots of them, and each of them is a promising target for drug development, and each of them has the potential to make a difference. But if your house is flooding, at some point you're going to want to clean up all the mess, but your top priority is turning off the faucet. I nearly said tap, because I'm English. So we want to, anyway, one way or another, call it what you will, we want to stop the flood at its source. And the result of at least 2 decades of research has produced the first drug candidate capable of doing this thing, which we call gene silencing, or more accurately perhaps, Huntingtin lowering in human patients. The drug was developed by a Californian company called IONIS Pharmaceuticals. The drug is called IONIS-HTT-RX. It is an antisense oligonucleotide, which is to say it is a single strand of chemically modified DNA. So it's something like this blue construct here. And what it does is essentially it sticks to the message molecule for the Huntington gene and to no other message molecule, like this. And when it does so, it essentially waves a flag that says to the cell, please get rid of this message. We don't want this protein. And the cell obediently dissolves the message. The protein is never made, or much less of the protein is made. And as I say, this specific drug has been under development for over a decade, but at least 2 decades' work underpins this first human trial. The drug has been chemically modified by Ionis in a way that improves the spread of the drug through the nervous system. And when you inject the drug into mice, which are sick as a result of producing the mutant Huntington protein, the mice actually get better. So the mice improve clinically. And that improvement is quite sustained. And if you give it before the mice have symptoms, it postpones the onset of symptoms in those mice. And it's been tested for safety in multiple species before it was given to humans. In the brain, impressively, a single injection of this drug into the spinal fluid, the drug hangs around for about a month. But even more surprisingly, the protein is suppressed for up to 4 months. So, and this is Carl Johnson's Huntington holiday idea. The idea is that if you can make life a little bit easier on the cellular level by taking away some of that Huntington protein, it seems that actually the brain is pretty good at recovering once you make life a little bit easier. So the company, the sponsor, Ionis, hopes to reduce protein levels by at least 50% in the cortex, and hopefully by about 15% or even more in the striatum, which is the deep brain structure that's affected particularly early in HD. And if 50% doesn't sound like enough, I turn to the experiment that nature has already done, which is that it has given people with the HD mutation a range of different lengths of mutation. So some people have a big mutation, and some people have a smaller mutation that's still capable of causing HD. If your mutation is at the smaller end, you still get Huntington's disease, but you get a form that usually comes on much later in life and is usually much less severe, progresses more gradually, and you have, your onset is maybe in your 60s or 70s or even later, and you can sustain a very good quality of life for many years. And that experiment that nature has done shows that we don't need to get rid of Huntington protein altogether. We need to reduce the total amount of toxicity that protein causes in the brain in order to make a meaningful difference and give people a good quality of life, or hopefully prevent or forestall the onset of symptoms. So the one caveat here is that this is not a drug that can be given as a pill. It has to be given by injection into the spinal fluid, intrathecal injection. And if that sounds terrifying, well, that's fair enough. But actually, this is a technique that's routinely used in spinal anesthesia and in cancer chemotherapy for people who have things like leukemia. So it's well established. It's not a typical day in the park, but it's certainly known to be quite safe and tolerated. So the objectives of the trial are to figure out whether the drug is safe and well tolerated, try and establish how long it sticks around, and does it lower levels of the protein, and does it change other features of the disease. But mostly it's about safety. Patients, 36 of them, and these are the Yuri Gagarins of Huntington's disease, right? These are the people who step onto the launch pad knowing that what they're doing is really potentially dangerous to them, but something astonishing for the rest of humanity. I can't express how much respect and admiration I have for these patients. It's happening in the UK and Canada and Germany, not the US, not yet. And 3 out of 4 patients get the active drug. Everybody gets 4 injections of the drug into the spinal fluid, 1 month apart, and then 4 months of follow-up. And it's being led by my mentor, Professor Sarah Tabrizi at the UCL Huntington's Disease Center, and sponsored by Ionis. So it's an ascending dose trial. Every few months, the dose is reviewed by an independent safety committee, and if the drug is safe and well tolerated, the dose is increased. And that has happened more than once without any concerns being identified. This is early September 2015, so 13 months ago. In Vancouver and in London, the first 2 doses here of the drug being given. I'm sorry that my hair is bad. it was a bit of a stressful week. But this is what it looks like when we inject this drug into the spinal fluid of our patients. Many patients have received 4 doses now of over 5 sites. There have been no safety issues so far. The trial has been described by a representative of the sponsor as blissfully quiet, which in a safety trial is absolutely the best that one can hope for. And it's projected to finish mid next year. Already, plans are underway as to what we do next. And Roche, which has bought the rights to take the drug forward if the results from this trial look good, is already thinking about what the next trial will look like so that if it's looking good, we can proceed without delay. There are other things happening as well. So this is the first ever human gene silencing trial for Huntington's disease. But actually, this whole field is really taking off. Everyone has 2 copies. Everyone who has the HD mutation has 2 copies, and almost everyone inherits a normal or wild-type copy from one parent and a mutant copy from another parent. And that mutant copy is the one that produces the mutant Huntington protein, the bad guy. The other copy, in almost all patients, produces the healthy protein, the good guy, which does useful stuff. So actually, if the current IONIS trial works well, that's great, but actually what would be theoretically ideal is if we can just get rid of the mutant protein, right, and keep the healthy protein chugging along. And a newcomer to the field, Wave Life Sciences, is aiming to do just that with a similar ASO drug with different chemistry to try and just selectively switch off the mutant protein. And zooming out even further, I put a little thing in. So they use mirror image molecules called stereoisomers to try and get the best possible attributes of the drug, so they optimize the drug. These are mirror image drugs, so this is a picture of Samuel L. Jackson, and the chemists among you will recognize that the mirror image is called Samuel D. Jackson. Okay, I think a few of you remember your high school chemistry. So that's the technology that Wave is using to try and optimize their molecules. There are several other companies attempting to silence the message, and at least one company, Shire Pharmaceuticals, is trying to use ways of interfering at the DNA level to try and reduce the production of the mutant protein. So it's actually a very busy field. It's a field in which there are incredibly sophisticated new ideas and a lot of innovation. And I think that's a testament, apart from anything else, to the ability of the HD community globally to organize and prove itself to be really good at recruiting and running clinical trials, which we've done for the past 20 years. So one other thing which is super exciting, which I discovered at the HDF meeting in Boston, is that some of these technologies require viruses to deliver them to the brain. Usually when you inject one of these harmless viruses, you get coverage of a small amount of the brain. This is a mouse brain. People have been mutating and evolving these viruses in a way that aims to improve the spread throughout the brain. And Ben Deverman's lab has produced a virus that can cover almost the whole brain with viral delivery of a drug. And this is hugely promising for future trials of these virally delivered treatments, which could be a single-shot treatment that would protect a patient for the whole life against the Huntington protein. So that's my whistle-stop tour of the field, and this is how it makes me feel. From Henry VI, Part 3, a little gale will soon disperse that cloud and blow it to the source from whence it came. The very beams will dry those vapors up. For every cloud engenders not a storm. I think that's something for us all to look forward to. Thank you.
I think what we'll do is we have 3 presentations, and we'll take questions for all 3 at the end.